Greetings, everyone, and welcome to the 6th and final edition of the Game Changer series of the policy webinars, AMR policy webinars. My name is Satyajit Sarkar, Satya for short. I'm the project coordinator for the RADAAR project and also the technical lead for the policy and advocacy part of our project. I will be your MC for today's webinar. Now, some of you would have joined new and some of you are you from before. So the overarching theme of the Game Changer series of policy webinars was to explore innovative ways of leveraging policy relevant data to drive AMR policy. Now, all the 5 webinars that have gone past, we had some excellent feedback from the participants. All the satisfaction ratings, somewhere in the 80s and 90s. So we're very grateful for that. So thank you for doing that. And also, while we were originally focused on the 22 Fleming Fund priority countries, which are 22 in number, we had registrants from over 50 countries and participants usually from 40 plus countries. So that was very heartening as well. Now, some really gifted minds working on AMR have been offering very critical reflections and very provocative thoughts through these webinars from a range of disciplines and perspectives. And all the webinars were moderated also by some really good experts, AMR global experts, absolutely. And the topics range from animal health, human health, environmental health, the social sciences, and this final one is looking forward-looking, blue skies, looking at innovative approaches, disruptive approaches using artificial intelligence and machine learning for AMR. And we have 2 very, very bright and fabulous speakers, very young speakers compared to the previous ones, which is just tells you which is the way to go. Now, each speaker and webinar attempted to answer overall 5 key sort of policy questions through their own disciplinary lenses. That is, in terms of AMR, where are we today? Where do we need to go? How do we get there? What works and how much it's going to cost? So that is the basic core of the questions that people have been trying to answer. I'll just go through the running order and the agenda for today. So this is the brief introduction I'm giving you, followed by the talk from Dr. Jonathan Stokes. He's based in Canada right now, and his topic is machine learning for antibiotic discovery. This will be followed by a brief Q&A between Jonathan and Dr. John Stelling, our much beloved and respected moderator. Everyone in the AMR world knows him. He's one of our consortium partner members, and I'll tell you about him in a bit. This will be followed by a talk by Dr. Brigham, learning the language of viral evolution and escape. He's from Stanford University. This will again be followed by a brief Q&A and discussion between John Stelling and Brigham. And then we'll have a general Q&A session, which will be open to all the participants, which will be moderated by John Stelling. And John will then wrap it up and give us some 3 to 5 key takeaways from this webinar. And this will be followed by exactly 30-second or 1-minute participant feedback and evaluation. And it's great to have all of you here. And I'd just like to give a brief introduction about John Stelling. So John Stelling is the co-director of the WHO Collaborating Center for Surveillance of Antimicrobial Resistance, and he's based at the Brigham and Women's Hospital in Boston. The focus of his work has been to support public health infrastructure and laboratory services, and he was for 3 years as a medical officer at the WHO Antimicrobial Resistance Monitoring Unit in Geneva. And since 1989, he's developed, disseminated, and support a piece of software which is called WHONet, and that is currently used to support surveillance activities in 120 countries and in over 2,300 laboratories in hospitals, public health, food, and veterinary laboratories all over the world. So with that, and without further ado, I hand it over to John. John, over to you. Thanks.
My great pleasure. To start off, at the bottom of your screen, you will see one option called Q&A and another option called chat. So you could use either, but we recommend that you try to put your chat questions into the Q&A box. And then some of those will be sent to me later for discussion. So we welcome your questions and your input. Okay, good. So subject of today is disruptive methodologies, artificial intelligence, machine learning, and AMR. When people hear the terms artificial intelligence, machine learning, forecasting, data mining, often just the concept of a black box comes to mind. You know, there's some box, you put something in, you put data in, and you get something out of it. But there's a lot of mystery as to what is going on in the black box. Many of our favorite movies or best known movies have to do with the subject of artificial intelligence in a good way or in a bad way. Currently, we have these mini series of Dune, Isaac Asimov's Foundation, Dan Brown, who wrote Da Vinci Code, also wrote Origin about artificial intelligence. One of the most famous, Arnold Schwarzenegger in Terminator. One of my favorite from history was Spencer Tracy and Katharine Hepburn were in the movie Discussant, all about computers and the value that they offer, but also you need to understand what they are doing. So when we think of data mining, we often don't really know exactly what's going on, artificial intelligence, but we need to keep in mind that things can go from the futuristic, the theoretical, the conceptual to commonplace very quickly. When you type in an address into an Uber or for a smartphone to get a car to pick you up, when you go to Google Translate, when you go to the supermarket and wave your food items over the scanner, these are very easy technologies to use, but we forget how complex the methodologies and algorithms there are behind it. So that's the goal of today, is basically to look to the future. And also, at some point, a lot of these things are going to be commonplace and it will have demonstrated their worth. In the laboratories, we now have MALDI-TOF for doing organism identification. It's easy to use, but you forget the sophistication of all the work that went into that. So for today, don't be afraid of these algorithms and artificial intelligence, but we do need to understand what they are doing, their strengths, their weaknesses, their applications, their relevance to what we personally do in our own work. So I invite you today, we're going to peek into the black box. In there are the algorithms, but there are also the people behind them, the insights, the testing, the evaluation, understanding of the limitations, and then close the black box so that eventually we can start to utilize these going forward. So our first speaker is Jonathan Stokes. He completed a PhD in antimicrobial chemical biology at MacMaster University, Canada. And then he came here to Boston, Cambridge as a postdoc at MIT and the MIT Harvard Broad Institute. He has recently returned to MacMaster to establish a research laboratory there. His work focuses on understanding the relationships between antibiotic structure, bacterial cell physiology, and the extracellular environment. Through dissecting these complex interactions, he aims to develop potent therapies with precision activity against bacterial pathogens while avoiding damage to the native microbiota. This has the potential to improve patient outcomes from antibiotic therapy while simultaneously decreasing the global dissemination of resistance determinants. His research areas include genetics and molecular biology, immunology, and microbiology and biochemistry. He is the lead author of A Deep Learning Approach to Antibiotics Discovery, a paper which found that neural networks can be trained to identify and predict molecules with antibacterial activity. So as a reminder, please put your questions into the Q&A box. I invite Jonathan to take over. I look forward to the presentation.
Thanks, John, for that really kind introduction. My name is also John. I'm an assistant professor in the Department of Biochemistry and Biomedical Sciences at MacMaster University. And like John was saying, a lot of my research focuses on discovering new structural and functional classes of antibiotics in an attempt to overcome existing resistance determinants. Right, so let's get this going. So, I'm sure we've all seen this at one point or another, right? Here's a timeline of antibiotic discovery, more or less, and we can see, right, that the vast majority of our productivity came between the 1940s and the 1960s, right? This is the so-called golden era of antibiotic discovery. And it's this period that's been characterized by high productivity in discovering different structural classes and functional classes of antibiotics, largely through mining secondary metabolites produced by microbes that live in soil, right? However, though, by the middle of the 1960s, right, finding new antibiotics through this approach became increasingly difficult. We ran into something called the dereplication problem. right? We kept discovering the same antibiotics over and over and over again, right? So in an attempt to overcome some of these failures in more recent decades, we've turned to high throughput screening of synthetic chemical libraries. Unfortunately, over the number of decades that we've been using high throughput screening to try to find new antibiotics, we've found 0 new clinically used antibiotics. Right? So it's been, an objective failure. Right? So, the problem is, like, this in and of itself, this failure in discovering new antibiotics wouldn't, if it was an isolated problem, it wouldn't necessarily be a huge issue, right? But it doesn't exist in a vacuum. What occurs in parallel with our failures in discovering new antibiotics is the global dissemination of resistance, right? So without immediate productivity, I would say, in discovering new antibiotics, we're on track to reach something like 10 million deaths per year by 2050, and that surpasses even cancer, right? So what we need then are new methods to discover novel antibiotics more rapidly and ideally less expensively than we have been in order to outrun the global dissemination of resistance, right? And that's what I hope ML has the capacity to help us with. So let's walk through this a little bit, okay? So if we look at a conventional small molecule screen, this is kind of what it looks like, right? We can screen hundreds of thousands to maybe a couple million molecules in the laboratory at the largest scales, right? What it looks like, let's say we screen our million whatever molecules, we'll have some low hit rate, we'll reconfirm, and then we'll chase the molecules in which we're interested in, right? However, this has a couple drawbacks, right? One, it's really, really expensive because you have to curate all the compounds you want to test. Right? And if you're curating a million molecule library, that's tens of millions of dollars more. Okay. Two, is it slow? Even though it's high throughput screening, it still requires a significant time investment in order to gather good data and analyze it. Right? And three, even though we're screening, a million molecules or 2 million, 3 million sometimes, right? And that sounds like a lot. Oftentimes, the chemical space that's explored in some of these synthetic chemical libraries isn't as vast as we would like, right? Now, let's compare and contrast this conventional small molecule approach with a machine learning aided approach, let's call it, right? Well, it starts off by gathering a training data set, right? So this is data that we use to train our model. And we can get away with training a model on, thousands to, let's say, tens of thousands of molecules, right? So this is orders of magnitude less than chemical screening, like conventional chemical screening at the largest scales. All right, so let's say we do that. Let's say we gather 10,000 chemicals worth of training data, then we can train a model, and then we use that model to run predictions in small scale and make sure it all looks good. Right? And then what we can do is take our finalized I like this model, and then we can run predictions on vast chemical libraries in silico, right? 10 to the 8, 10 to the 9 molecules. This is orders of magnitude more than we could ever even think of screening in a laboratory, right? And in doing this, right, we can save costs because we're training on a much smaller chemical set. It's faster because we can run predictions in silico like orders of magnitude quicker than we can screen them in the laboratory. And three, because we can run predictions on such vast chemical sets, the physical chemical space that we can explore is much greater, right? So that's the theory behind, leveraging contemporary ML approaches to help us discover new antibiotics, right? Okay, so let's look at a little bit of data. How do we actually do this? Okay, so this is a screen of 2,500 compounds against E. coli. All we're doing is looking at the inhibition of the growth of E. coli in liquid media in the lab, the simplest screen we can do, right? So this is growth, chemical number, right? And then all we did is every dot here is a different compound, right? What we said is everything in blue, we're going to label with a 0, and we're not going to call it a hit, right? And then everything in red, we're going to label it a 1, and we're going to call it a hit. Why? Well, because everything in red inhibited the growth of E. coli in this experiment. Everything in blue either kind of inhibited or didn't inhibit, right? So this, where we drew our line to inhibit where we drew like our hit cutoff, right, is kind of arbitrary, but for us, this worked fine, right? So everything that did not inhibit the growth of E. coli is labeled with a 0. Everything that did inhibit the growth of E. coli is labeled with a 1. Great. Okay. So what we want to do then is we want to train a model to learn the relationship between all of these structures and the corresponding label, right? So how do we do that? Well, we're gonna go into a little bit of technical stuff, but it's not going to be crazy, right? So let's say we have this molecule, right? Let's say this is just some molecule from our training data set. It doesn't matter, right? What we want to do is encode the structure of this molecule in a way that can be used to train a property prediction model, right? We want our model to be able to predict whether this thing is antibacterial or not, right? So we can build an informative molecular representation of this, right, by teaching every atom in the molecule about every other atom in the molecule, right? We can teach the molecule about itself. Okay, so what does that actually look like? So as a concrete example, let's say we have this bond here, right? 2,1. What we want to do is teach bond 2,1 about all the bonds that feed into it, right? So this would be bond 4,2 and 3,2. They both feed into 2,1, right? Okay, bond 2,1 is represented by this vector, right? This is it's just a 3-dimensional vector, 3 numbers, right? Now, what we want to do is we're going to take the vector that represents 3,2 and the vector that represents 4,2. Here's the vector that represents 3,2. Here's the vector that represents 4,2. We're going to just add them together, right? So this is the sum of vector 3,2 and vector 4,2, right? We're then going to take this sum vector and then we're going to concatenate it, right, with the original vector that represented bond 2,1, right? So we're just going to stick this vector onto the original vector that represented bond 2,1. We're then going to run that through a feed-forward neural network, and this is going to result in an updated vector for bond 2,1. And this updated vector, right, now contains information about 2,1 as well as 3,2 and 4,2, right? This is called a message passing neural network because what we've done is we've taken information from 3,2 and 4,2 and fed it into 2,1. So what that means now is that this bond 2,1 understands itself a little bit better in the context of its local molecular neighborhood, right? Bond 2,1 now knows about 3,2 and 4,2. It knows a bit more about what they look like and what they're doing, right? So you can imagine if you do this message passing step over and over and over again, right, what you're going to end up with is a single vector that captures the complexity of the entire molecule, right? We're going to end up with a vector that knows the entire molecule, that captures what's in it, right? And it's this final molecular representation that we use to make property predictions, in our case, whether some molecule is antibacterial or not, right? Cool. So that's all we did, right? So we trained our message passing neural network on 2,500 molecules, whatever, right? On these structure property pairs. Structure, it did not inhibit the growth. Structure, it did inhibit the growth, right? And we trained our model, and what that allowed us to do was show our model a brand new structure it had never seen before, right? And then based on that structure and based on the weights in the trained model, it would spit out a number between 0 and 1. If it was closer to 0, the model says, I don't think this compound is antibacterial. If it's closer to 1, it says, I think it is likely to be antibacterial, right? Cool. It's not super complicated, right? And then, so that's exactly what we did. So we trained our model, and then we ran predictions. And we ran predictions on a bunch of different chemical libraries, but the one I'm going to focus on here is this library called the Drug Repurposing Hub at the Broad Institute. So this is a chemical repurposing hub, right? At the time, it had about 6,000 chemicals in it, right? So what are we looking at here? Let's look here, and what we see is the prediction score of all the chemicals in this library, right, that were made by our model, right? And we can see, if we rank order all of the predictions, right, that some molecules have really high prediction scores, but the vast majority have really low prediction scores. And that's exactly what we would expect, right? the drug repurposing hub is not full of antibiotics. So if we had a whole bunch of molecules with a really high prediction score, something would be broken, right? But the fact that we see a relatively small proportion of molecules having really high prediction scores is aligned with what our initial hypothesis would be, namely that this library is not full of antibiotics. Okay, cool. So these predictions are fine, but they don't mean very much unless we validate them in the laboratory, right? Here's where the importance of wet lab work comes into play. How good is this model in the real world, right? Okay, so what we did is we took the 100 molecules with the highest prediction scores, and we just tested them in the lab. We said, of the 100 highest predictions, which ones work in the lab? How good was this model? Well, we tested the top. We ended up creating the top 99 predictions. But when we tested them in the laboratory, this is again just against E. coli. We're looking at growth and the number of predicted molecules, and we observed that 51.5% of them, so 51 out of the 99, inhibited the growth of E. coli sufficiently that we would call it a hit, right? And then we asked, well, out of these, 51 chemicals that actually inhibit the growth of E. coli, right? Which ones are structurally different from known antibiotics, right? Because that's ultimately what we're after, right? We don't want to find analogs of stuff that we already have, right? We don't want to find analogs of like existing cephalosporins or aminoglycosides or whatever. We want to find structurally new stuff, right? So what we asked was, of these 51 compounds, which of these are structurally distinct from known antibiotics? And it turns out that there was one compound in this set that was structurally different from known antibiotics, and it was this compound that we decided to name halicin, right? So we can see, so Tanimoto similarity is a calculation of how structurally similar this molecule is to other molecules from our training data set, right? So we can see the Tanimoto similarity. And Tanimoto similarity, for a little bit of background, the higher the Tanimoto similarity, the structurally closer it is. So if you have two molecules that have a Tanimoto similarity of 1, they're the same molecule. If you have two molecules that have a Tanimoto similarity of 0.1, they're very, very different. They're not the same molecule. They're completely different molecules, right? So we can see that this molecule halicin, the Tanimoto nearest neighbor from our training data set, was below 0.4. Those were a family of antiparasitic compounds. And the Tanimoto nearest antibiotic from our training data set was an antibiotic called metronidazole, and the Tanimoto similarity between halicin and metronidazole was about 0.2, so very, very different. Okay, so we had this molecule, and then we just ran some in vitro tests with it, right? First thing we did, this is growth as a function of halicin concentration. We observed that the minimum inhibitory concentration, MIC, of halicin against E. coli is 2 micrograms per ml. This puts it in the ballpark of everything, a lot of other antibiotics that work against E. coli. Okay. We then asked whether halicin was bacteriostatic or bactericidal, right? Does it inhibit the growth of E. coli or does it kill cells? Well, we observed that with higher concentrations of halicin for prolonged durations of incubation, that we lost cell viability. This is viability as a function of concentration, therefore it is bactericidal, right? It kills cells. Third, given that the structure of halicin is different from known antibiotics, we wanted to test halicin against E. coli harboring a range of different resistance mechanisms, right? So this is lab strain E. coli harboring different resistance genes on a plasmid, and we observe that halicin retains activity in the presence or absence of these different resistance enzymes. And that's aligned with what we would expect based on the structural similarity of halicin to known antibiotics, right? And then last, this is an experiment that I really like. So I'm interested in resistance and antibiotic tolerance, right? So I asked, does halicin have activity against antibiotic tolerant persister cells? So in this experiment, what we're looking at is viability as a function of time, okay? In dark blue, we just have E. coli growing in media. There's nothing fancy here. This is just E. coli growing. In light blue here, we have E. coli treated with ampicillin. And what we can see is that ampicillin kills some cells, but then it stops killing, right? And these cells are antibiotic tolerant, right? But what we did an experiment where we where we followed what we treated with ampicillin, got these antibiotic tolerant cells, and then we treated these tolerant populations with halicin, and we observed that with increasing concentrations of halicin, we were able to kill more and more and more of these antibiotic tolerant persister cells. To me, this is really exciting. Antibiotic tolerance is an understudied phenotype, and the fact that halicin did have this unexpected activity, I just thought it was really unique and exciting. So this is fine and good. It's all in lab strain E. coli. The next question that we have, obviously, is does this work? Does this molecule work in clinically relevant stuff, right? So we curated each bar is a different clinical strain of CRE, Acinetobacter baumannii, or Pseudomonas aeruginosa. There are 36 different clinical isolates per bug here. We observed, I'll summarize this. Halicin worked against carbapenem-resistant Enterobacteriaceae and Acinetobacter baumannii, but did not work against Pseudomonas aeruginosa very well. And it's likely that halicin didn't work against Pseudomonas aeruginosa due to impermeability, right? We know that the cell surface of Pseudomonas is really difficult to get molecules into, but 2 out of the 3 worst gram-negative pathogens I would consider is not bad. We were still happy. So then we wanted to elucidate mechanism of action, right? So we tried the first thing we tried to do to elucidate mechanism was a resistance evolution experiment. So in liquid media, we tried to evolve resistance to halicin over 30 days. We were unable to do so. And furthermore, we tried this in an evolution experiment on solid media. And again, we were unable to isolate resistant colonies. And we can contrast both of these results with our ability to readily evolve resistance to ciprofloxacin, right? Ciprofloxacin, big fat colonies. And in liquid media, we were able to evolve resistance quite readily as well. So failing, the quick and dirty experiment of evolving resistance and then sequencing the genome to find the resistance mechanism that would lead us to mechanism, we turned to RNA sequencing. So this is just E. coli treated with halicin, and then we did an RNA-seq experiment. What we observed was the immediate and significant downregulation of genes involved in locomotion and flagellar biosynthesis, right? And it turns out that this is a common transcriptional response when cells are starved for ATP. Now, I'm not going to bore you with the details, but we did a whole bunch of different experiments. Here's a fluorometric experiment. And what we ended up understanding was that halicin resulted in bacterial cell death and indeed this transcriptional response because it was dissipating the proton gradient that exists across the cell surface, right? So in E. coli, on the cytoplasmic membrane, you have a high concentrations of protons on the outside of the cell and a low concentration of protons on the inside of the cell. And this proton gradient is essential for many, many cell processes, right? And it turns out that halicin was actually dissipating this gradient. And we can talk a little bit more about that if you want to go into the details. So that was fine. Interesting mechanism of action, difficult to evolve resistance. So the last thing we did was check to see whether halicin worked in mouse models. And indeed, we observed that halicin in a topical Acinetobacter baumannii skin infection model was able to clear the infection in about 25 hours. And in a C. difficile gut infection model, halicin was able to eradicate C. difficile from murine feces more rapidly than the vehicle or a metronidazole control, right? So this was kind of like our first, entry into the application of ML for antibiotic discovery, and we're continuing to grow this effort specifically in the context of narrow spectrum antibiotic discovery. So we have now models trained across a bunch of different pathogens. Here are 4, for example, Acinetobacter, E. coli, Pseudomonas, and Staph. And we have molecules that are predicted to have activity against every combination of these 4 species, right? But because we're interested in narrow spectrum compounds, we're interested in these 1541 that are predicted to have anti-staph activity, these 889 that are predicted to have anti-pseudomonal activity, and so on and so forth, right? We're diving into these data now, and I'll give you a sneak peek into one thing that we're working on really diligently. Here is a molecule that has anti-Acinetobacter activity. This is I'm not going to show the structure because we're still working on it, but we've recently found this molecule. You can see that it only has activity against Acinetobacter baumannii, but not against CRE, Pseudomonas, or S. aureus. Each dot is a different clinical isolate. And what we're looking at on the vertical axis is bacterial growth, and on the horizontal axis is different concentrations of this molecule relative to the MIC of the molecule in lab strain Acinetobacter, right? So here we're looking at probably 70 or so clinical isolates, and here is about 40, 40, and 40, right? And we can see that with increasing concentrations of this molecule against Acinetobacter, we can inhibit the growth of all of these isolates, but with high concentrations of this molecule against CRE, Pseudomonas, and Staph isolates, we don't have any growth inhibition. And even more excitingly, I would argue, is data down here. So this is a heat map showing the growth inhibition of 27 different commensal isolates, right? So with narrow spectrum antibiotics, we don't want to eradicate all of the good microbes that live in our gut. And indeed, a whole bunch of conventional, commonly used classes of antibiotics do, right? They're not specific. They kill both the pathogen and all of our good bugs, right? And that's shown. Growth inhibition here is represented in red, right? But if we look at our compound at an MIC concentration, see how everything here is green? That means that our compound did not inhibit the growth of any of these common commensal species, right? So we're still studying this. We're looking at mechanism of action. We're going into mouse models of infection next week. So stay tuned for that. Okay, I've gone a little bit over. I'd like to thank all of these folks here for funding this work, collaborators from various labs in Canada and the Boston area. And these are three of my students who worked on the data that I summarized in the last two slides. So that's it. Thank you for paying attention.
Thank you, John, for that. In the interest of time, I'll ask, I have one comment and one question. The comment is, in part, it highlights that there's not one magic algorithm that can do everything. Each algorithm has certain strengths and weaknesses in certain applications. For example, algorithms to look for, sometimes you do want drugs that are similar to ones that exist with perhaps better pharmacological profiles. Sometimes you want a new target site. Sometimes you just want to see what's out there. So different algorithms will have their strengths and weaknesses, and these need to work together. That's sort of a general comment. So my question is, in traditional drug detection, we go out into the nature, we find a bunch of plants and products and soil samples, and we look for chemicals. Yeah. What you have described is a way to make this process much more efficient, to try to highlight those chemicals that merit further investigation. Mm-hmm. But then we end up with the new bottleneck is still afterwards is about toxicity profiles. You know, so many drugs have been discovered that were very promising, and then 5 years later you look it up and it got stuck at some point. Yep. Toxicity, ineffective, and one very good thing for antibiotics, but it's also a problem for new drug development, is that antibiotics are very safe drugs by and large. They're targeting primarily bacterial processes that we don't have, or fungal processes we don't have. So sometimes you might have a very good drug that still gets taken off the market because there's this question mark of toxicity. Yep. Some drugs like gadifloxacin, gemifloxacin, were evaluated much more thoroughly than the older ones, the ciprofloxacin, but they were taken off the market because of questions over the toxicity profile. So what you've described can allow a lot of new drugs to be put into this slow process of clinical trials, animal models, et cetera. Could you just comment on any work that might be going underway in data mining, machine learning, that might help this next step in getting a promising chemical to actual clinical trials? Yeah, so it's super. I'm glad you brought that up, because so our lab was working on a different project looking for antifungals, right? And fungal biology is so much closer to human biology that this is a problem that we're constantly thinking about. So in an attempt, again, this is like our kind of first entry into trying to build models that can predict toxicity. So we have two sets of screens going on. We're looking for antifungals against our fungal pathogen, and we're also screening a whole bunch of human cell lines in parallel, right, on the same set of chemicals. And the idea, at least our hope, is that we'll be able to train relatively robust toxicity prediction models to be able to predict whether some new molecule, let's talk about an antibiotic, some new antibacterial molecule is also going to display toxicity
in vitro, at least against a whole bunch of different human cell lines. Now, the question mark around that approach is, how well does in vitro toxicity translate to in vivo toxicity in a mouse or in a human or whatever? It might be relatively low, but if it's better than no model, it's still progress worth pursuing, right? Because, empirically, it would be very difficult, I think, to gather sufficient data in order to train a reasonable model to be able to predict toxicity in a mouse, let alone a human, right? Now, you can think about mining existing databases for this data, right? The problem that I kind of would have with that is, like, how similar are all these different models, or are all these different data sets, right? That already exist looking at the toxicity of some molecule in some model system, right? So we're having all these different data that are gathered different ways, and we're going to stick it all together and try to train a model. Is that model going to be worth anything? What's it going to learn? I don't know. And that's what scares me about that approach. But, it might be something that we try, like, on the evenings and weekends. I don't know. But we are actively thinking about this problem.
Yes, it's important that different groups are working on these different elements. They all need to come together because the science for each of them and the needs are different for validation and robustness, et cetera. Okay, thank you. And there are some questions, but we'll raise those afterwards for the group Q&A after. So I'd like to proceed now to introduce Brian He. So Brian did his PhD in computer science and artificial intelligence at MIT. He is currently a Stanford Science Fellow at Stanford University School of Medicine, where he develops algorithms and machine learning methods with a focus on biological application. He completed his PhD, as I mentioned, at MIT and was an undergraduate at Stanford. He has also worked on machine learning for early pipeline moonshots at Google X and for health-related applications at Illumina, for whole genome sequencing, et cetera, next generation sequencing. He is the first author of Learning the Language of Viral Evolution and Escape. As a reminder, please put your questions into the Zoom Q&A area. If you put them in the chat, we'll also see them, but by preference into the Zoom Q&A area.
So today I'll be talking about forward-looking research. This is a kind of a line of research and kind of predicting the evolution of pathogens, and it's interdisciplinary in that we're leveraging these algorithms called neural language models that were originally developed in the context of natural language to instead start to learn something about the rules or the language of protein evolution instead. And so this is work with a fantastic set of co-authors, Bonnie, Brian, Peter, Kevin, and Ellen, done across 2 institutions, MIT, where I was a graduate student, and Stanford School of Medicine, where I'm currently a fellow. So to begin, I just want to start out with this very basic question in biology, which is how predictable is evolution? So on one end of the spectrum is this thought experiment by Gould, where he imagines rerunning the evolution of life on Earth. And according to Gould, it's so random and determined by environment and just random chance that if you reran that process, you get completely different organisms, completely fantastic creatures. But on the other end of the spectrum is this idea by Morris that actually evolution is so constrained by these internal intrinsic factors that if you reran that simulation, you'd get an organism that looked like humans or something very close to humans. And actually there's really a lot of predictability within evolution. And so this research is trying to push the boundaries of evolutionary predictability, and in particular motivated by the problem of escape from immunity or from drugs. So the majority of this talk will be on trying to predict the rules and maybe future evolution of viruses. And then in the latter half of the talk, I'll extend these ideas beyond viral proteins to other tasks as well. But to begin, this is the problem of viral escape. I'm sure all of you are probably aware of this. So if you have some original virus and the host develops an immune response to this virus, say a neutralizing antibody that binds to the viral surface protein, what the pathogen can do is actually mutate in successive generations the surface protein so that this neutralizing antibody no longer binds. And then this viral particle is free to infect host cells. And so this is the problem of escape from say, antibody-mediated immunity. And viral escape is a very big problem. It's the reason why you need a yearly flu shot, and that flu shot might not be very effective depending on the year. And it's the reason why we have no effective vaccine for HIV despite decades of development. And so I believe that if we're going to have any effective vaccine, especially for HIV, we need to start learning the rules of how these pathogens mutate themselves to evade immunity. And of course, we're in the middle of a global pandemic, and there's definitely questions about how long the current vaccine-mediated immunity against SARS-CoV-2 will last. It looks like it's holding up pretty well for now, but that could change at any time in the future. So how do we start to get a handle on predicting or at least learning the rules of escape? So we started from basically like a first principles approach, and we had this key initial insight, and that small changes can have big semantic effects. So what do I mean by this? So for example, say we have this English language sentence, the boy pats the dog. So I can actually make a single character change to the sentence that drastically alters its meaning or its semantics.
Brian, would you, do you want to move on to the next slide? I'm still on the title slide.
Oh, okay, hold on. Uh-oh. Let's see. Is this okay now? Or is it?
Yes, single residue changes enable escape. Thank you.
Okay. So, yes, viral escape. This is the problem of escape. The viral protein mutates itself to evade, say, antibody binding. It's a big problem for for vaccine development, and potentially for SARS-CoV-2 as well. And say we have the sentence, the boy pats the dog. I can make a single character change that drastically alters the meaning of the sentence, this P to an E mutation. Hopefully you're caught up on that intro and the minute summary. Okay. And so we have this key insight that small changes can have big semantic effects. And likewise for viral escape, it's been shown that even a single residue change, a mutation to this viral protein sequence can induce escape from broadly neutralizing antibodies. So for example, if I mutate a histidine to a serine in this influenza hemagglutinin protein, then this broadly neutralizing antibody no longer binds. So how are we thinking about connecting these two ideas? We see that like maybe there's some connection between small changes having big effects on an overall sequence. And so this is the key idea that we kind of contributed to kind of learning the language of viral escape. So say you have this initial immune response corresponding to this initial viral surface protein, and corresponds to the sentence, the boy pats the dog. So if I'm this virus and I want to escape from this antibody immune pressure, what I don't want to do is I don't want to mutate my surface protein so that it misfolds or that it loses the ability to infect cells. I want to maintain some notion of validity or grammaticality of the mutation. But what I also don't want to do is I don't want to mutate the surface protein so it just looks similar to the immune system and this antibody can recognize me. I want something that not only preserves infectivity or validity, but also induces high antigenic change so that this antibody no longer binds. And we're kind of thinking about this idea of antigenic change as a semantic change. So preserving, say, the grammaticality of a sequence while inducing high functional or semantic change. And we kind of formulate this problem. We call it a constrained semantic change search, or CSCS. It's basically like a word game. You're given a sequence of tokens from some language, and your goal is to find the single token change that induces the largest semantic change to the sequence, but is constrained by the rules and the grammar of that language. this problem is given in generality, this could be a sequence of words from an English sentence or a sequence of amino acids from a viral protein. So in terms of computational algorithms for doing this, how do you make a model that can perform this constrained semantic change search? And this is the idea of a computational language model. So all a language model is a way to assign a probability to a given sequence. That's all it is. And the way a lot of language models work is that they predict the probability of a missing word or missing token given some sequence context. So for example, this sentence, the American president blank to Japan yesterday, a language model would predict the probability for each word in the English language of it appearing in this blank. So maybe the American president went to Japan yesterday. This is, a high probability of 0.5. Maybe a rare word like the American president absconded to Japan yesterday might be a little lower. And then non-grammatical words like xylophone would be given 0 probability because they'll never appear in this blank. And similarly, we can do this for amino acid sequences, and we can train a neural network model similar to the models that John was talking about earlier, that takes some sequence context as input and predicts the probability of some missing amino acid or some missing word in the sequence. And when we train a language model on thousands or even millions of valid sequences, so these are grammatical sentences or valid amino acid sequences, these language models can start to extract the patterns of which words go together in what context. And that will start to correlate with grammaticality or validity. So this is all a little abstract, but we actually trained a language model to do this CSCS problem on news headlines. I'll briefly go through some examples. So for example, this original headline, Australian dead in Bali. So according to the language model, the closest single token change that was the semantically closest to the sentence is Aussie dead in Bali, so pretty synonymous. But the CSCS proposed change is Australian ballet in Bali. So all of a sudden, this headline is a lot more positive, depending on your opinion of ballet, of course. And you can also imagine this being, a valid headline, Australian ballet in Bali. But it's semantically different, even though it's off by only a single word. Some other example changes: blast off of Apollo 8 turns into blast victims of Apollo 8. This is already a strange headline to begin with. Excuse me, you left a gorilla suit on the bus. Turns into, excuse me, you left a gorilla killer on the bus. A lot more negative in this. And then you can do this all day. I'll give you one more example. Wine growers revel in good season turns into wine growers revel in flu season. So hopefully this gives you an idea of this problem. A single token change that preserves grammaticality, but induces a high semantic change in the sequence. So this is somewhat of a toy problem in the natural language case, but what we actually wanted to do is predict a viral escape. And so to do this, we can train these language models on these large sequence databases of viral proteins. And I know some of you are involved in policy and data curation across a large number of countries. And so I would just want to say that this work would not have been possible without these international efforts to maintain these databases and collect the actual data through viral surveillance efforts. So that work definitely contributes to the ability to train these large language models on existing viral sequence data. And so we train this language model on these databases, a separate language model for influenza, or a language model for HIV, or a language model for SARS-CoV-2. And we want to see if the language model can learn these patterns that correspond to escape. And so to validate the predictions, we can use these laboratory experiments. For example, this experiment done by Jesse Bloom's group at the Fred Hutch, where you can make all amino acid mutations to, say, influenza HA that are compatible with viral growth, and then subject some cells to a viral infection with and without the presence of some immune selection. So say, neutralizing sera from someone who had influenza. And then you can just deep sequence these cells to see which viruses made it into these cells. And this will, when you compare the immune selection to the control, this will allow you to determine the mutations that actually contributed to escape from this antibody while preserving infectivity. And so what you're seeing here in this plot is each point corresponds to a single amino acid change to, in this case, influenza hemagglutinin from an H3N2 strain. And they're plotted on the x-axis according to grammaticality, and on the y-axis according to semantic change. And these experimentally validated escape mutations are drawn with these red X's. And you can see that they exclusively cluster in this high grammaticality and high semantic change portion of this portion of this landscape, this like top right quadrant here. And I would just like to emphasize that this language model had no idea about protein biochemistry or escape. It was just trained on these unlabeled amino acid sequences, and it had to learn these ideas of fitness and antigenic change just directly from sequence variation alone. So we thought this was an extremely promising result. When we look at the predicted escape patterns on the structure of, let's say, hemagglutinin, they correspond to known sites that have high, that are known to have high escape probability. So in the case of influenza, it's the head region of this protein versus the stalk. We made these predictions for also SARS-CoV-2 spike, and in particular the N-terminal domain and the receptor binding domain as being especially escape prone, and these were confirmed after we made the predictions by a number of the scientific field, basically. And so I know probably a lot of you are wondering about SARS-CoV-2, and so in this kind of follow-up work led by Cyrus Mayer at Vir Biotechnologies, we found that language model grammaticality can predict mutations that arise in different waves of COVID-19 up to 4 months in advance. And as kind of an illustration of the value of being able to predict these escape mutations, one of these mutations, S494P, that was highlighted by the language model and also other evolutionary metrics, actually does, in some cases, completely ablate neutralization of, in this case, one of the FDA approved monoclonal antibodies for COVID-19, but it doesn't for other antibodies, including the one made by Vir. And so potentially you could design these antibodies or select antibodies from patients with this escape, these escape predictions in mind. And so this is kind of the dream of, or why you would want to try to anticipate these escape mutations. So in the last 5 minutes, I just want to briefly describe how these ideas can extend outward from viruses to other proteins as well, and beyond immune escape to other problems like drug resistance. And so we know we can predict local evolution with these language models, but a big question is, evolution often involves more than single residue mutations. So what do you do in the case of recombination or highly mutable proteins? And so we have this key insight that more global complex patterns across an entire fitness landscape can be approximated by local predictions made by a language model. And this is conceptually similar to ideas of fitness landscape going back all the way to Sewell Wright in the 30s and kind of the early days of evolutionary biology. And we can also now scale up these language models so that they learn generic patterns in protein evolution across all known proteins in say, UniProt. So all sequences that have been observed across all species may be filtered by sequence similarity. And this, in particular, we use a language model from the Facebook AI protein team trained on 3 million sequences from UniRef50. So when we apply this language model to kind of predict these evolutionary changes, we can actually see this is a 2D visualization of the landscape of the evolution of a influenza protein, the nuclear protein, and these kind of stream arrows. This is not wind flowing over 2D landscape. but it's actually the flow of evolution across this sequence landscape. And surprisingly, we found that the flow of evolution reconstructed the known temporal evolution of this influenza structural protein. And in particular, we can also look at the roots of this flow in order to actually reconstruct the known zoonotic crossover events in influenza. So these major pandemic, the major points in which these pandemics began, we can actually backtrace from our predictions to identify them. And we get a good correlation between our predicted evolutionary order and kind of the real temporal evolution of this influenza protein. These ideas also surprisingly generalize across generic proteins. I won't spend too much time, but this is the flow of evolution across the landscape of cytochrome C. It recapitulates the order of taxonomic categories as observed in the fossil record, and this applies to the longest trajectories of evolution as well, spanning all 3 domains of life. Okay, so I wanted to conclude just looking forward and hinting at potential directions that you could take this research. And in particular, our model was trained on, or the original CSCS model was trained or sold as predicting viral escape, but actually this this approach can also be used to also predict mutations to drug resistance, especially if those mutations are very common in a population that's sequenced. So this very talented master's student at MIT, Andy Cho, showed that you can also predict mutations arising in HIV or tuberculosis using similar approaches. And a final thing I want to close with is now that we have kind of some additional ability to predict evolution and kind of harness the power of evolution ourselves, we can do laboratory directed evolution, but guided by these large scale language models with an unprecedented knowledge of evolutionary biology. And so we're currently working on directing evolution using these approaches and maybe evolving the protein therapeutics in a way that kind of competes with the evolution of viruses in nature, for example. Some key takeaways are that language models have the potential to improve our ability to model evolution, predict evolution. Of course, there's a lot of environmental factors that aren't captured by this model, but we show state-of-the-art ability to predict evolution. Sufficient training data is important. So thank you to everyone who has collated this data and collected sequence data to train these models. And of course, a successful implementation of these models will require not only interdisciplinary collaboration within academia from natural language, computer science, and biologists, but also with policymakers as well. There's a lot of things I skipped over in these studies. Here are some references where we can go and learn about these projects in much greater detail. And I'd just like to again thank my co-authors on this project, Ellen, Brian, and Bonnie at MIT, Kevin, who's at Microsoft Research, and then Peter, who's in Stanford Biochemistry.
Great, thank you for the presentation. Also have one comment and one question, and we encourage everyone to submit questions either by chat through the Q&A, or then I will also open it up so people can just raise their hand, and then you can just ask the question directly. Thank you for that. It's one of the things that when I see these things is it's good to know what is possible and how we can understand what is possible. But as you pointed out, it's still unpredictable. If everything started all over again, would we end up with humans or dinosaurs or something that looks completely different? Which brings me to my question is how can we possibly connect this to policy? Also, I love movies. The movie Time After Tomorrow, they say, well, global climate change may destroy everything in 10 years. Oh, I'm sorry, I mean next week. No, I'm sorry, I mean tomorrow. The thing changes very quickly, of course, movie. So an article a couple of days ago about potential supervolcano eruption in Yellowstone National Park. Might be tomorrow, might be in 10 million years. So these things can happen, will happen with a great degree of predictability. But at some point we have to tie this to policy. And this is very clear in the case of COVID. So I think a lot of decisions, unfortunately, by politicians a year and a half ago were based on COVID as much as we knew at that time, presuming it was not going to change. So a number of people were sort of favoring this idea of herd immunity. And herd immunity, there's herd immunity if the organism doesn't change, and herd immunity if the organism does change, and the epidemiology is going to be extremely different. And as of course we have seen with Delta, the versions before Delta, now the new Delta Plus, if there were a great appreciation of mutation and the escape that you were describing, hopefully it could have led to a more stringent response instead of just let it pass and then it will be over. With these mutations, even if people have been infected, it's still not going to be over. So any thought about how you can tie some of the kind of work you've been involved with to actually helping to inform the policy? Assuming, knowing that there's all this uncertainty. I remember with the mad cow, bovine spongiform encephalopathy, there was an estimate from Oxford saying, I don't remember the numbers, but somewhere between 6 people and 6 million people might die in the next 30 years. So, but we still have to make policy on the basis of these uncertainties. So any thoughts, any comments?
Yeah, especially going, forward into the future, like the greater the time horizon, the greater the uncertainty. So I think there's like different things you can do in the near term and in the long term. So we actually worked, we teamed up with this team at the CDC out of Fort Collins to kind of use like a computational method like this to screen. They have a bunch of, they're connected to a bunch of laboratories around the United States that are sequencing SARS-CoV-2 genomes. And so they need an initial filter to filter a lot of these sequences for anything that's like phenotypically interesting that they can then phenotype using, say, more standard neutralization assays. So I think that's one immediate area in which you can maybe have a week or 2 weeks or a month in advance knowledge of which new variants are likely to start evading, say, vaccine induced immunity. And then maybe a longer term is the if you can design a therapeutic that potentially in at least predicted by a model is able to withstand pathogen evolution that's predicted to go like a year or 2 years or 3 years in the future, even though there's a lot of uncertainty there. Potentially a therapeutic could be able to withstand multiple mutational paths going even forward into the future.
It'd be interesting to connect in things like virulence and pathogenicity, dissemination capability. Right now, we often just have to wait and see, is it more virulent, is it more transmissible? But if we could do data mining to say, well, this particular modification may predispose to an increased rate of aerosolized transmission, for example. Also, a lot of these human factors, I used to work with David Heyman in Geneva, and a lot of his early work in the 1970s was with Ebola. It was one thing to have small outbreaks of Ebola in remote villages in Central Africa with very little movement of the human population. And now with great movement of food and animals and people, a lot of the old forecasting, all it takes is one flight, and all of a sudden what could have been historically contained is now existing all over the world. So John, I'd like to invite so yeah, Brian, feel free to make a comment, but John, if you could just join the video chat again, that would be great. Yeah, Brian?
Yeah, just a brief comment. I think, and what we did with the people from VIR is actually incorporate epidemiological information into the predictions and show that, it definitely really helps with predicting, virulence and stuff like that. But again, yeah, predicting the future, it's there's a lot of environmental considerations. If everyone just social distanced for a month, then there would be no evolution of this virus. But there are things, given some assumptions on evolutionary processes that are predictable.
So John, the first question is for you in the chat box, but do you have any comments before we get to that? Any observations, or just dive into the question?
No, it's funny because Brian and we had a conversation a couple months ago. Was it a couple months ago? We were talking about whether we could apply his, evolution prediction models to predict antibiotic resistance against some like novel compound. And I forget, what happened? It was, I think our conclusion, Brian, was like the training data doesn't exist because there are like insufficient sequence data for antibiotic resistance in the context of like some specific scaffold, right, or something like that.
Yeah, definitely. I think the kind of the advantage of viruses is that there's a lot of surveillance data available with which to train the models, and it seems to be less common for antibiotic resistance. And the results that we had for tuberculosis were only possible because of private data set owned by a large government that was able to train that we used to train those models. But in terms of public data sets, there's some resources, but I don't think the sequence complexity is enough.
And it's a tricky problem too, right? Because it's not like every mutation that confers resistance is a SNP, right? It's like you can have like horizontally acquired genes and pumps and all kinds of stuff, right? So like the complexity of the mutational space, if you want to call it that can confer resistance is a lot bigger than like an amino acid shift, right?
Yeah. And then in bacteria, you have like, the viruses are kind of simple machines. Like bacteria, you have like intergenic epistates. Like you potentially have a compensatory mutation.
Oh yeah,
a completely different gene.
That's what I was thinking about, John, while Brian was answering all the questions.
Thank you. So, John, the first question is for you. It's from Alexandra Costa from UNICEF, I believe. John, you said you were looking for molecules that were structurally different from known antibiotics. But if you are training your neural network with a library of molecular patterns extracted from known antimicrobials, wouldn't you expect a bias in the selection of molecules that are similar to known antibiotics?
Yeah, that's the whole. So yes, you're 100% right. Like the model will learn very well the chemical space on which it was trained, right? And if we trained it on a whole bunch of antibiotics, which we did in our training data set, we had the known universe of antibiotics, right? It learned what a fluoroquinolone was really, really well, and it learned what a cephalosporin was, and it knew what tetracyclines were, all this stuff. But what these like contemporary neural approaches allow us to do is something called generalize, right? And in my opinion, that's kind of the most important feature of these like new deep learning models is they are able to generalize to new chemical spaces reasonably well. And that's why, we continue to use them over more conventional like machine learning approaches, right? That perhaps don't generalize into new chemical spaces very well in the context of molecular property prediction. So that's kind of like, we talk about it in the context of tension, right? So I'll explain what that is. So let's say, we have a model and it was trained on this, right? And here's like the chemical space and all of the molecules in this chemical space. That's what our model was trained on. But I'm sitting in a room and here's the chemical space on which we're running predictions, and it's all around me, and this phone is small and this room is big, right? What happens is, as you get further and further and further away from this chemical space on which it was trained, right? The predictions become, they tend to go down and they tend to become less certain, right? There's more uncertainty around every prediction. But this is the tension that we talk about. It's like, how far away from our training data set can we get and still be relatively sure that the predictions that are being made are real, right? And that for us anyway, we largely empirically determine that. We're lucky that we have a whole bunch of, chemists and biologists and robots and stuff around us. We can empirically validate all these predictions, right? So we look for instances that where we get far enough away from our training data set that it's a new molecule and it's predicted to do the thing we want it to do, and it still works when we go and empirically test it in the lab, right? But you're right, you caught on like something that like, it's like a fundamental feature that we always think about. And it's like this trade-off between like generalizability and having the predictions actually work when you go and test them in the room.
Also reminds me on a different subject, but after the 2001 anthrax attack, there was a very large concern about bioterrorism. And not only that, there was SARS, pandemic flu, avian influenza. So there's a lot of funds and activity and research put into outbreak detection algorithms. And there was a large focus on syndromic surveillance, looking at things like cough, over-the-counter purchase of diarrheals, rash, because if you're not looking for anthrax, you're not going to find it. So the idea was to try to bring in syndromic surveillance data. And there was a society called the International Society of Syndromic Disease Surveillance. The URL was syndromic.org. And after the first few years, a lot of that great excitement sort of faded because it was finding things people already knew about. There was a lot of noise. And there were just problems with the underlying data, but the mathematics lasted. So they just reapplied a lot of that mathematics in the models to other data sources like confirmed laboratory data, confirmed diagnoses. So this is an example where you want to develop the algorithms, the approaches, the tools, and then apply them to different data sets. So you've applied these to certain data sets, but a lot of these algorithms could be applied to other data sets as they become available. So with Syndromic, they changed the URL from syndromic.org to something else. They changed the name of the society from the International Society of Syndromic Disease Surveillance to simply the International Society of Disease Surveillance. But a lot of the algorithms that were developed early continue to grow and to be used in unexpected new directions. So with all these algorithms, you start somewhere and you learn, and then you try to find useful applications for them. Okay, great. So Brian, the next question is for you. When an escape is predicted, can the evolutionary trend be interrupted, or the only solution is to prepare for the new potential emergence variant? In other words, what is the best response to a prediction?
Yeah, that's a good question. I think I'm definitely not like a person who works in policy, so I can just comment on maybe the degrees of a prediction. So when you get a computational prediction and it's predicted to have high escape, I think the first thing you need to do is experimentally validate it against say neutralizing sera or something like from someone who's received like one of the vaccines, and see if it actually in a neutralization assay confers escape. And you can also maybe try to measure infectivity as well using cell lines, but then that will inform for future decisions. And then maybe if the epidemiological data suggests that this variant is taking off in certain parts of the world, I think then you should start considering policy changes like shutdowns and stuff. But.
Thank you. John, I have two questions for you. By training your model on a small sample, what is the chance that you will miss effective new molecules because your model scope was limited in scope? Or is that not the case? Another question, very broad. If everything goes well, approximately how long will it take for a new drug like this, Allison, I presume, to reach market level?
Okay, so the first question, so I think I'm inherently a pessimist. So I think that most of what I do is wrong, and the probability that we're missing really good antibiotics in any chemical set on which we run predictions is super high, right? And it's, here's like the numbers game I like to play with myself. So, John, you'll probably appreciate this. So like the total number of drug-like chemicals that could theoretically exist is on the order of 10 to the power of 60. Right, which is like a number that's like you can't even begin to fathom it. It's like more grains. It's like more than there are grains of sand on the planet. You know what I mean? It's just like some crazy number. So it's like if we train a model on like tens of like let's say like we screen like every chemical that we can buy in the world. What we're probably looking at like 10 million or something, 20 million in that, like that's the order of magnitude. And we'll train like the best bunch of the best collection of models we can, and then we'll run predictions on the totality of drug-looking chemicals that could ever exist, and then the probability that we're going to still miss good drugs is really high, right? But this, so that's like my pessimistic, nihilistic answer. But it's a different question when it comes to in reality, what are we trying to do? We're trying to discover new antibiotics, right? And then so people, when they get sick, we can make them better, right? So there's this idea of what is good enough, right? So instead of trying to chase perfection and get a model that can accurately make predictions on all of the potential antibiotics that are theoretically possible, let's train a model that can work well enough to get us a compound that has legs that we can eventually turn into a drug. So it's not letting perfection get in the way of fine, right?
Well, before the second question, just want to comment on this part. For me, I'm an optimist in the sense that there's not one global solution that's going to solve everything. The question is, does each innovation bring us ahead in some areas? What is the strength of this? What have we benefit from this? Is one thing going to come out or two things going to come out? Has there been some benefit? Is this the full solution? No, we need other algorithms, other data sets, other approaches. So you don't do the one and, the expression, all of your eggs in one basket. You do this because there's value to it, but that's not, you just don't stop there. One group's team or other group's team need to pick up, identify the strengths of this, but the weaknesses of it as well. In my own work, we do a lot of work with statistical outbreak detection, use the free SAT-scan software for space and time scan analysis. And we published several articles about the value of our algorithms in large hospitals. And then when we started going to small hospitals, we were not finding important outbreaks. It's a numbers game. So does that invalidate what we did? No, it doesn't, but it qualifies it that we found a good tool that has certain value to some people, but we need to continue our work, change the algorithm, change the baselines, change the settings, change the thresholds. So there's not one magic formula that's going to solve everything.
No, I mean, like ML, I mean, I don't think it's a panacea. It has to be used in parallel with all of like the conventional wet lab and whatever other approaches that we've tried, right? So that's why it's still important for people to be looking at like exotic secondary metabolites from odd microbes for potential compounds that could be antibacterial, right? That's like a lot of the work that, just down the hall from me, Jerry Wright does, right? He's finding interesting chemicals, interesting secondary metabolites every day. Nathan McGarvey next door to me as well, same thing, right? But yeah, it's going to be a combination of all of these different approaches, these computational approaches and biochemical approaches, microbiology, that all come together. And then we're all going to make different progress at different rates. And then eventually we'll start discovering new antibiotics, right? My favorite is the application of these ML approaches. I think it's the coolest, or else I wouldn't do it, right? And I think it is going to result in a lot of productivity in the relatively short term. Again, that's why I do it. If I didn't think it was true, I'd quit. And now for the second question, how long does it take to make a drug? The canonical time horizon is 10 years, right? I'm hoping that we can do this a lot faster than that. So a lot of the molecules that I found and continue to find. So right now we have a nonprofit that's set up to attempt to accelerate the rate at which these compounds can get into through the IND process into phase 1. If I'm going to be like a hyper optimist, we can maybe shave a few years off of that 10-year horizon. But still, there's fundamental efficacy and safety hurdles that we have to clear in humans, right? And being able to predict what these molecules are going to do in humans, before running the experiment, it's we're just not there yet, right? But I mean, again, this goes I grew up playing a lot of hockey. I'm Canadian, right? So this is all shots on goal, right? So how many good shots on goal can we get before we start making real progress to get molecules eventually approved and into patients, right? And
you were just earlier this idea of data mining to also help on some of these things clinical pharmacological profiles, toxicity profiles. So the work you're doing, I think, can get products to this point much more quickly. You still have the other points, and then if there are innovations of AI or other things, there are other steps that will continue to be bottlenecks that are demanding new solutions.
I think it's a question of uniform data availability. That's what I'll call it. A ton of this clinical trial data and stuff like this. This all exists, right? But a lot of it's housed within pharma companies who don't make it public, right? So it'd be nice if there was a global sort of consortium that could curate all of this existing human human clinical toxicity data sets and disseminate it to people who are doing drug discovery programs, right? And then we could be in a better position to potentially train models that could predict with some reasonable accuracy whether some new molecule is likely to display some form of human toxicity when it goes into a patient. You know what I mean?
And there's certainly a global effort in bioinformatics to get genome sequences available. The question is, will there be similar efforts by pharmaceutical companies to share what is a global good, but it's also proprietary information?
Yeah, and I mean, and then like, and I understand like why there's kind of like, you know, we're at this, we're kind of fighting each other here. It's like, you know, there's like public good, but if I'm looking at this from the perspective of a drug company, I know I also have to keep investors happy, right? So it's like, what, like how, like they're kind of like incompatible. You know what I mean?
We have to keep in mind that many pharmaceutical companies have left, the large ones have left antimicrobial development because it's high risk with, it's often low benefit with high risk. There's a request from Satya to make a verbal comment. So Satya, go ahead.
Yeah. Okay, so this has been a fascinating discussion, especially this latter part of the Q&A was very nice for me. What I wanted to ask was just taking a different step in terms of, then it's a question to all three of you, John, including yourself. That is, what is the message that we should be giving to low and middle income countries? The approaches that were discussed here today, is that something that low and middle income countries or developing countries, as they are sometimes called, should they be leapfrogging into this area? Is this an area that they should start investing within their own countries? Or is that still some time away and they should stick to, shall we say, conventional approaches? I mean, it's sort of a broad question like that. What is the message that we can give for these kinds of approaches for low and middle income countries? Thanks.
I'll give a very brief comment in my area of work for statistical outbreak detection with routine laboratory data. For me, the answer to that is definitely yes. If your laboratory data suggests there's some outbreak using, whether you call it artificial intelligence, we're using algorithms to find potential outbreaks. So for something like that, I would definitely recommend yes. We've demonstrated value there. Regarding some of these other comments, I invite either one of you, both of you, to make some comments.
Brian, you go first.
Oh, I mean, yeah. So I think definitely like, well, in terms of the forecasting, like if an algorithm is developed, you could easily just run it and apply it globally. But you know, in terms of the overall area of machine learning, I think it's in some way it's like it's very accessible to just anyone to kind of learn the underlying tools and techniques in machine learning in a way that, like, say, doing like a big drug screen is not. So, but and then in terms of data generation, I think like a lot of the it's really important that these data sets are not biased towards certain geographies in the world, and so I think like global surveillance and not biasing the data towards more resourced institutions. Especially in training these models is definitely extremely valuable.
Yeah, I think, so for what I do, the trickiest part is gathering the training data with which to train the models, right? So once this data exists, it's not private. I'm not hiding it. We publish it, when we write papers. So this data exists in the public literature. And I think it's to everyone's benefit. The more people who are playing with these data sets and looking at all of the things that we miss, it's all going to contribute to, the eventual discovery and development of new antibiotics or new whatever, infectious disease thing, let's say you happen to be interested in, right? So yeah, the application of machine learning I think everyone should be doing, right? And I feel like the barriers to entry now are like sufficiently low that, there should be a global community of, interested and invested people leveraging these approaches to solve, in our case, antibiotic resistance, right?
Also, I'm going to, in one of my takeaways, I'll come back to this point. Brian and John have mentioned some applications of machine language, artificial intelligence. There are many other applications that, of course, were not covered. I think many of those would also be very interesting for healthcare facilities and governments to consider, especially as it relates to clinical decision support, policy, guidelines and things. Okay, so great. So I'm going to just give some wrap-up messages, and I'll turn it back to Satya. So I was asked to prepare 5 key takeaways, and I'd like to format these better. I'll write them up a little bit better later, but these are transformative, I think, in disruptive technologies. You've heard some examples that there will be many dead ends or misdirections, but there will also be unanticipated directions, applications, successes. These will come in part from experience, but in part from clever people applying these things in ways that other people had never thought about. Even dead ends will highlight the need for new directions or problems with existing data. I was talking with somebody recently about 3D printing. He said, we can make anything now. They said, well, what are you making that couldn't be made in other ways? He said, well, nothing really right now. There's not a demand for that. Somebody will think of demands, but he said, we mostly use it to do things that we could have made manually, but more efficiently and to specifications. But they said, we're not transforming the actual products that we make. Okay, that's number one. Number two, there are some directions that should be of greater value for the management of resistance. Drug design, you've heard about phenotypic and genotypic outbreak detection, response and modeling, clinical decision support at a high level. Also, there's this idea of clinical decision support for individualized medicine. There's the US Precision Medicines Initiative, where the idea is maybe at point of care to prescribe this particular drug for this individual because of their genetics, their HLA factors, their personal history, their risk factors. So personalized medicine would benefit from this. There's a project called Gideon. I met the founder of it about 25 years ago in Israel. Gideon, Global Infectious Disease Epidemiology, something network, which was based on a lot of old-fashioned things and valuable. I have patients with these fever and disease and travel, predict what disease the patient may have, especially for tropical diseases. Now they do the same thing, but a lot of it is driven by data mining approaches to help predicting what the oddball, unexpected tropical disease might be. Point number 3, bioinformatics, genomics, moving more from phenotypic monitoring to genotypic. Not only the sequences, but the 3-dimensional structures, of these potential drugs, the 3-dimensional structures of the target sites. We first discovered penicillin, and then we discovered its mechanism of action was the beta-lactam ring was mimicking D-alanine at the end of the growing peptidoglycan chain. Can we start doing anything in reverse order where we have a 3D target site? Can we go back and try to develop things that might be able to target it? That's number 3. Number 4, to recognize the limitation of the black box approach. The thing is, people just trust machines, but they're not aware of their biases, the limitations. It might be very good for certain things, like for me, for the large hospitals. It doesn't work for a lot of our small hospitals with the large data volume. You have these enormous confidence intervals. And then you always have unexpected things. The financial collapse of 2008, a lot of that was driven by a lot of these artificial intelligence machine algorithms that had certain assumptions. Once those assumptions got thrown out the door, the algorithms drove a lot of very bad automatic decisions. There were other things going on as well, but that was among them. And then finally, basically, what are the key applications of artificial intelligence that facilitate, that help facilitate and national policymakers that they should be exploring now, touching on Satya's point? How do we communicate findings to policymakers? How do we highlight the limitations and strengths? What are some of the biggest areas where some of these things could be applied in the next 5 years? And I'll just leave it at that. Thank you, and I'll hand it back to Satya. And also my thanks. Oh, I do have one final question for John. Hal, is that related all to Hal from Space Odyssey? I'm a big movie fan. Yeah, Hal is, people who might know, is the name of the computer. HAL is followed in the alphabet by IBM, but the authors deny any connection. So it is from Hal. Okay, thank you.
Very good. That was a clever one. Okay, thank you. Thank you very much, Brian and Jonathan. It was fascinating. It was quite technical, I must say, but interesting nevertheless. And since we will have the advantage of looking at the videos again, so maybe I will look at it again and try to understand better. It's worth knowing that, but really tremendously, it was really nice of you, and it was moderated beautifully by John, bringing in other experiences and acting like a bit of a devil's advocate, which is nice. So I thank you all. And I would just request that all participants who are attending right now to just hold on for 30 seconds more and just fill up this simple poll. You'll have to scroll down. So there are just 4 questions, and we would love it if you could give us your feedback on this. How do you rate the overall webinar, this one? How satisfied were you with the content and reflections of the speakers? Was this helpful in acquiring new knowledge? And will you recommend the webinar series to others? Of course, the webinar series is over, but these are also going to be available on YouTube, and all of you will definitely be receiving the recordings and the presentations wherever we have got consent from. And while you're filling that up, I'd just like to also recognize that we had some media people in this webinar and earlier ones also, but there are 2 people. One is a person called Andil from East Swatini and Millicent from Nigeria. Both these journalists, we really thank them because they attended all 5 webinars and the subjects were completely different. The only thing holding together was AMR. So we would really like to thank them for their participation throughout all the 5 webinars. So with that, I would like to thank everyone who participated, and of course, our wonderful team here who's backing me up, who are all behind the scenes workers. Jiong, who's our platform manager, Shailin, Prerna, Anthony, Jinui, who are all working on this together to make this happen. And we will make all these, like I said, the videos and the presentations available. And the recordings of all the previous ones are also there. And with World Antibiotic Awareness Week coming up, there's also a major call to action on AMR by Wellcome Trust and UNICEF and a whole bunch of other people. They have also set up a facility by which these materials can be shared, and many more people will be able to see it. So thank you, Brian. Thank you, Jonathan. And thank you very much, John Stelling. And we will be in touch. Okay, thank you very much.
Thank you, everyone.